everyone, and welcome to the Laboratory Testing and Automation virtual event. My name is Rebecca Pompano, and I'm an assistant professor at the University of Virginia. And I'm going to be sharing with you today some new technology to combine the fluidic control of microfluidics with the power of intact live tissue samples. I want to remind you that you should feel free to submit questions at any time during the presentation, and I'll follow up with you via email. So please be interactive. Where I'd like to start is by reminding everyone that your tissues in your body are highly spatially organized. And what this means is that the cells and molecules that make up your tissues are arranged so that they're touching each other in specific ways. And this, of course, enables them to have functions that they wouldn't have if they were all blended up together. And what you're seeing on the screen are 3 images from 3 different organs: the lymph node, the brain, and an ominous looking tumor. And what we notice is that there's cells clustered together. There are fluids passing through that we can't see, but they would be carrying any secreted proteins or small molecules that are released from one cell and then carried to neighboring cells in order to have paracrine signaling. You can imagine that if you took a brain sample and crushed it up and homogenized it, that it would no longer have the same functions that it has when it is in your head. And yet, often that is how most in vitro experiments are done. So what we see is that people take cells that have been blended, and then they're either plated onto a flat plastic plate or suspended into a 3D hydrogel culture, usually in a homogeneous manner, occasionally with some spatial organization that's forced into the system. And although this provides a lot of experimental control, it doesn't enable you to ask questions about how the tissue is organized or how cells contacting each other in the way that they did in vivo leads to function. On the other hand, if you have an in vivo experiment, such as in a mouse or a higher animal, you are able to preserve all of that spatial structure, but you lose the ability to drug the tissue of interest at will or easily to be able to make measurements. And so we really need a hybrid method of looking at spatial structure in tissues. There's a number of people pursuing different ways to do this, including spatially patterned cell cultures. But here, what I would like to share with you is an alternative approach that a few labs have been developing, and that is to combine the power of ex vivo culture, meaning samples of tissue outside the body, with microfluidics to control the environment. So here, just to walk you through the advantages and disadvantages of working in a live sample of tissue, I'm showing you a table with the 3 different modes of experimentation at the top. And then if we think about what are the features of working in vivo, it's that intact spatial organization. That's preserved if you're going to work in slices, but not if you're working in cell cultures. On the other hand, if you're working in slices, you lose the connectivity of the tissue to other organs. So for example, if you're studying the brain, that brain is no longer connected to the gut or the lungs once you are working with a slice. That could be either an advantage or a disadvantage, depending on the questions that you would like to ask. On the other hand, with slices, you're able to access substructures of tissue and organs that would have been deep within the animal and therefore not accessible to live 2-photon imaging or other analytical methods. And so you're able to bring out some parts of tissue that you wouldn't be able to study in vivo. On the flip side, there are features of working in slices that are more akin to what you get in a cell culture dish, such as the ability to repeatedly analyze the same sample without having to do an endpoint experiment for every time point, and the ability to deliver drugs at a defined concentration and time within the dish or the chamber that you're culturing the tissue in. We can also easily collect supernatant in order to do downstream offline analysis of secreted proteins or molecules. And we can use simple wide field imaging modalities instead of having to use sophisticated 2-photon microscopy. So by working in slices, we sort of gain most of the benefits of in vivo work and cell culture work, but it is a slightly higher level of complexity than regular cell culture work, and so you do have to get yourself set up for that. So the way that these samples are prepared is by collecting the organ of interest from either a human biopsy sample or from an animal donor. And then if it's a small piece of tissue, it's usually embedded in a clear agarose gel for physical support. And what you're seeing on the screen in the center picture are 3 samples of, in this case, murine lymph node. So lymph nodes from a mouse, they're a few millimeters in diameter, and they're embedded into agarose gel. And then those are sliced on an instrument called a vibrotome. This is different than a cryotome or a microtome because the tissues are still alive. That means that they're soft. They haven't been fixed or frozen. And so the instrument is set to slice several hundred micron thick slabs of tissue. And that ensures that only the top and bottom faces are damaged by cutting, while the center is still a healthy, vibrant piece of tissue. So, of course, this is a lab automation talk, and so we're not going to talk just about tissue. We want to put it into a piece of technology that comes from the field of microfluidics. And microfluidics is perfectly suited to study these tissue samples because they're really at the larger end of what a microfluidic chamber can accommodate. So when you're working with tissues, they're often a millimeter in diameter or perhaps a few hundred microns in diameter. And then that leaves you with all of the lower range of microfluidic length scales, like tens or hundreds of microns, to control the environment of the tissue. So we're able to, for example, deliver streams of fluid locally to subunits of tissue or to connect multiple tissues together. And those are things I'm going to share with you later in the talk. The combination of ex vivo slices and microfluidics has been around for perhaps 15 years, and it really began with brain slices. That makes sense because brain was the first organ that was really sliced frequently by neuroscientists, and for decades now they have been collecting and culturing tissue slices, either acute for just a few hours to study the electrochemistry, or even for months in order to study development of the brain. And microfluidics engineers quickly figured out that by using fluidic control at the microscale, they could more efficiently control nutrient and waste exchange, and they could control the gas environment around the tissue, often in a way that was more controlled than in classic cell culture dishes. But we can go far beyond that. By using microfluidics, you can deliver to just one part of the tissue, shown there in that orange circle, while preserving the ability to interface the device with imaging and other analytical modalities. And so this type of technology has been applied by a handful of groups around the world. This is still a small field, and it would be exciting if some of you listening today decided to join this area. And so just to give you a sense of what is possible, I'm showing you 4 different types of applications that have been pursued so far. We see some drug and toxicity testing in the upper left that came out of Sebastian Verporte's group in the Netherlands back in 2010, where they actually connected multiple types of organs together. I believe it was liver and gut in order to look at metabolic changes in response to drugs. We've also had mechanistic experiments where microchips that were interfaced with electrochemical arrays were used to map out signaling in the brain. There's applications in personalized medicine, for example, taking patient tumor samples and putting them onto a chip in order to test different combinations of chemotherapy, and that came from the Gervais lab in Montreal. And then, interestingly, a 6-month-long culture of testis tissue is shown in the lower right, and they were actually able to get spermatogenesis and then breed mice using the sperm that were produced. So this was highly viable tissue culture for over 6 months, and that came out of Japan just a couple of years ago. And so you can see that there's a lot of potential for using this type of technology and combining the intact tissue with its spatial organization with the fluidics. What my lab works on is taking this technology and using it to build new tools for the study of the immune system. This is an area where there's a lot of need for new tools in the field of immunology, especially ones that look at spatial structure, as most of the advances in immunology lately have come from either single cell analyses or flow cytometry or in vivo imaging, where there isn't much control over what's happening in the tissue at any given time, and it's mostly watching through a window. And so by combining microfluidics and chemistry and immunology, we hope to develop new tools so that we can ask questions about how your immune system is organized and how that drives the spatial effects or how that drives the response in an immune event. So just to very briefly orient you on the tissue that you're going to see featured in the rest of the talk, but then we'll get back to the technology. My lab really focuses on an organ called the lymph node, and you may be familiar with that organ. It's your tonsils, for example, which get swollen when you have an infection. And this is an organ that is highly structured. It's similar to a brain. Where there are substructures that are responsible for one function or another, such as antibody development or the development of killer cells. And they're scattered throughout your body in strategic locations so that you can respond appropriately to infections in different parts of your body. What my lab has been developing for the past few years is a method to collect the lymph nodes from mice and then slice those. And we've tested extensively the viability and the immune function of those tissues ex vivo in order to ensure that they can still respond to inflammatory stimuli or other immune stimuli. And we have found that a slice, shown, like the one shown here, still preserves the spatial organization that we would expect to see in vivo. What you're looking at is a piece of mouse lymph node that is alive in culture, and we've labeled in green where the B cells are, and in blue where the lymphatic vessels are. And we can see this beautiful structure is preserved ex vivo. Okay, so with this technology, what are some of the things that are possible? What I'm going to share with you are two short stories, one using tissue slices in fluidics to ask questions about local events in the tissue, and then the other to ask about interactions between multiple organs in the body. So two different levels of spatial organization, and we're going to start with local stimulation. Okay, so the question that really drove me when I started working on this several years ago was how do local events trigger functions in the body? And again, here we're talking about immunity, but this is just as real in other organs such as the brain or the gut or the lung that you may be interested in. And so we wanted to ask, what happens if you deliver a drug to one part of an organ versus another? For example, how quickly does it spread through the tissue, and does that vary by area of the organ? And then later, we might be able to ask questions about triggering events and propagation through tissue. But how do you deliver to just one part of a piece of tissue that is only millimeters in diameter? You can imagine that pipetting here is quite difficult, especially when this is in a bath of media. And we didn't want to insert any microneedles into the tissue, as that may damage it and cause there to be a reaction that changes the response. So we took a cue from some prior work out of David Eddington's lab at the University of Illinois at Chicago, where he had laid a microfluidic channel beneath a chamber holding a brain slice, and then used ports to deliver to just one part of the brain tissue, as illustrated here. And so what we did was we adapted this down for the lymph node, and you can see the device design here. So there are 3 layers to this microfluidic chip. The bottom layer has an array of channels. The middle layer has an array of ports where there's 1 port per channel. And then the upper layer is simply a culture chamber where you can lay your tissue. And so the concept is that you would choose a feature of the tissue that you want to deliver drug to. For example, maybe that red area of the tissue is interesting. And then you would choose the channel that has a port that terminates beneath that region and deliver drug to it, as shown there in orange. So we construct this device out of 3 layers. Right now, it's just made out of polydimethylsiloxane, or PDMS, using standard photolithography methods. And you see the image of the chip there on the right with its large central circular chamber where you can lay the tissue. And if we zoom in there, we can see the array of channels in the lower layer and the array of ports on top. And you see that the ports are scattered. That's to increase the chance that they fall under a region of interest in the tissue. So I want to show you what it looks like when we do local delivery. We have here a fluorescent image of a piece of tissue where it is laid down atop the array of ports, and I've outlined that tissue in a white dotted line. And then we're delivering a fluorescent probe through a channel beneath, and you can just barely see the top of that channel at the top of the image. And then we see the drug starting to come up through the port in this semi-radial fashion from the port. When we draw a line scan across the image, we find that these distributions tend to be pretty Gaussian, as you see in panel B. And then we can fit that Gaussian curve in order to find the area under the curve and the width of the curve. And that enables us to get a sense of how much are we delivering. And we see that if we deliver longer, we get a linear increase in the amount of dye that's in the tissue. And we get linear increases in the width of the pulse, at least for these short delivery times. Eventually, it will saturate the tissue. So we have pretty good control over delivering drugs to regions of the tissue. And you'll notice there in panel D, for the shortest pulse times, a few seconds, we can get resolution on the order of a couple hundreds of microns, depending on the size of the molecule. And so for these protein-sized molecules, we tend to have 200 or 300 micron resolution, and that's sufficient to target substructures of tissue, such as in the lymph nodes, say a B cell follicle or the T cell zone, or in the brain, you might be targeting part of a cortex, for example. We did some fluid modeling to understand whether these pulses are going all the way through the tissue. And so what you see here is a model of the cross section of the device. So there's the channel on the bottom with fluid flowing through it. You can see the arrows. And then on top is supposed to be a slab of tissue, which we modeled as a porous matrix. And what we've done here is we've flowed for 5 seconds a pulse of a drug, and the concentration of that drug is shown here in the color scale. And now we're starting to flow buffer in behind it. And so what we see is there's this ballooning up of a spot of drug pushing up through the tissue. And then if we wait 1 minute, we see that, with buffer coming in behind, it's gone up and out the top of the tissue and is beginning to be washed away. So the delivery is passing all the way through the tissue, and that's a feature of this design where we're not just stimulating the surface of the tissue, we're actually penetrating and getting all the way through the depth of this thick slab of tissue. Okay, so that was the first generation of this chip with these stationary ports beneath the tissue. One drawback of that chip design is that it means that if you lay down your tissue and you wanted to deliver somewhere that didn't happen to fall over one of the 10 ports, you would need to move the tissue manually in order to get it to align with a port. And that isn't too hard for a highly trained scientist, but for someone who might be new, or if you want to just have technicians doing this, you might damage your tissue by having to move it. So we had a vision of changing the chip design so that you could lay down the tissue and then move a port to be anywhere that you wanted it to be. And as you can imagine, you can't move anything in a solid block of silicone rubber. So this required a complete redesign of the chip. And I had a talented grad student who took this on, and so she created what is referred to as a slip chip design, where we have a piece of tissue lying atop an array of ports. At this time, we have over 80 ports beneath the tissue. And then that array, instead of being directly connected to a channel, is now floating on a layer of oil. Below that is an additional channel and then a single port. And I've shown that filled here with green. So what do you know about oil and water? They don't mix. So this is basically keeping completely separated the solution of drug that might be in that channel down below from the culture media that is surrounding the tissue. And then what you can do is slip and slide the 2 layers, since they're lubricated by this layer of oil, in order to connect the the port of interest above with the channel below, and then you can turn on a pump and deliver up, and now you have delivery akin to what we had in the other chip. And then we can disconnect again and turn off the pump in order to turn off the flow. And so my student Meg worked out all the physics of this. It's based on surface tension to keep the delivery fluid from leaking out into that oil-filled layer. And we found that we were able to deliver selectively to regions of interest repeatedly within the same tissues. And so we're continuing to work on this design to move completely away from ports and into more of a membrane format so that we can deliver truly anywhere on demand. Okay, so what might one do with a system like this where you can locally deliver? One of the first applications that we decided to target was to ask, how do molecules move through different parts of the tissue? Because we know that in tissue, you aren't diffusing in a simple way. Instead, you're experiencing what's called hindered diffusion, where molecules have to move through the tortuous spaces between cells. And this has consequences ranging from development and repair of organs to the paracrine signaling between cells, and importantly to drug delivery. As you think about a drug that has exited the bloodstream and needs to travel several hundred microns before it reaches, say, a tumor or a region of interest in the tissue, it's doing that by diffusion. And diffusion is currently measured in tissues by either using FRAP, fluorescence recovery after photobleaching, where you soak your entire tissue in a dye and then bleach one spot and watch as the molecule diffuses back in. That works well for inert probes, but won't work well for a bioactive molecule that might change the tissue during the soaking time. So instead, we wanted to use local delivery, and that is usually done by some sort of needle inserted into the tissue, and then imaging or electrochemistry to watch it spread. But again, we're worried with an immune tissue, and perhaps with other tissues as well, that inserting a needle that is tens or hundreds of microns in diameter into the tissue could cause damage that changes the transport behavior that we want to watch. So this was a great application for this gentle fluidic delivery. And so we took the device that I just shared with you, the simple one where everything is stationary, and we simplified it further. So we have just a single channel and port beneath the tissue. And the goal here is to deliver bioactive molecules into the tissue and then watch their spread. So a typical experiment looks like this. You see on the left an image of a piece of tissue that we've labeled the B cells, which tend to be around the edges, in green. And then we're delivering a red fluorescent protein. In this case, it's tumor necrosis factor alpha that we labeled with rhodamine dye. And then we deliver for 5 seconds, and you see that in that second image, which is labeled 0 minutes. So that becomes time 0, is that pulse of dye that's been delivered to the center. Then we just stop the pump and we watch over time as the molecule diffuses away. Then we can quantify by image analysis the spread of that molecule and back out the diffusion coefficient. And we validated this against FRAP in inert gels with standard probes, and found that we're within 2-fold of reported measurements. So we're fairly confident in these numbers. And then we were able to make the first measurements of the diffusion of TNF alpha and other bioactive proteins moving through different parts of the lymph node. And we were able to see that indeed in the tissue versus an inert gel, we do get slowing down of the transport of these molecules, presumably due to binding, which is what you would expect. Whereas an inert molecule like dextran, shown on the left, travels with the same diffusion coefficient through all of these materials. So this is sort of a demonstration that you can watch as molecules move through the tissue by using this sort of technology. And we envision that this has applications in things like drug delivery and studying paracrine signaling and propagation through tissue. And so if any of you have questions or ideas about that, I'd be happy to talk with you by email afterwards. Okay, so in the last few minutes, I'm just going to share briefly one other story, which is using microfluidics instead of looking at structures within tissue to begin to think about bodily level organization and multi-organ connection. So as you know, no one organ in your body operates in isolation. They're connected via the blood and lymphatic systems. And The organ pair that we were particularly interested in was the lymph node, of course, and in this case, connecting to a tumor, as tumor immunity is becoming a really exciting area where there's potential to take advantage of the immune system to fight off patients' tumors. And so it's known that tumors drain to the lymph nodes near them through lymphatic channels, and then cells and molecules from the lymph node can get back to the tumor via your bloodstream. And so we wanted to make a model of this in microfluidics in order to preserve the intact tissue with their spatial structure and that basic fluidic connection. And so my postdoc Sangjo Shim designed a chip that with this basic concept where you have a tumor and lymph node slice, each on the chip and connected in a one-way loop of fluid flow. In this case, for simplicity, we first put beneath each tissue a track-etched membrane that restricted cell movement by having small pores, and we were using 2-micron pore size membranes. And that is a design choice that can be varied in the future in order to test the effects of cells traveling, or, for example, to measure metastasis. So one of the important features of this chip design was to get the fluid flow to pass through the tissue, not just over it, because we want to pick up paracrine signals and carry them to the downstream tissue. And so this is a result from a model of fluid flow simulation in console multiphysics, and there's the tissue right there, so you can see it appearing and disappearing. And Sangjo found that by placing an inlet on the upper left and an outlet on the lower right, he was able to drive fluid flow pretty much straight down through that tissue so that he could pick up anything that was being secreted and carry it out. Other designs where the inlets were both on the same side or both on the top or both on the bottom really didn't provide this flow through. We fabricated this using standard PDMS microfluidics, once again with a 3-layer chip and a track-etched membrane that goes beneath the tissues in order to hold them in place and restrict cell movement. And then we needed to validate how proteins moved between slices and whether proteins secreted by one tissue could indeed be captured by another tissue. And so to do that, we set up a simple model system. We made mock tissues out of agarose and loaded them separately with either a fluorescent protein coupled to neutravidin, shown on the top, or biotinylated beads, shown on the bottom. So this is to model a receptor-ligand pair. And we could quantify the fluorescent intensity in those mock tissues before the experiment. And of course, only the protein-loaded slice has bright fluorescence. And then we cultured these overnight in the incubator with continuous fluid flow, the way that we would do for tissues. And we saw that indeed all of that fluorescent protein was captured by the receptors in the other tissue after 24 hours with minimal loss to the device. And that was achieved by blocking the channels in a particular way with BSA beforehand. And if you're curious about that, you can look in the paper. Okay, so finally, as a proof of concept, we had this chip that we knew could support 2 slices of gel or tissue and have proteins move between them. And so we asked, to what extent can this model some of the features of tumor immunity? And we started off with just one feature, which was that we know that in vivo, often the lymph nodes are immunosuppressed by the tumor, meaning that the T cells can no longer respond to really any type of stimulus very well. And so we asked to what extent that could be mimicked by the chip. So we did an experiment where we had a mouse this is carrying a 4T1 breast cancer tumor, and then we collected its lymph nodes, and we did a measure of the capacity of the T cells to respond to stimulus. We hit them with an anti-CD3 antibody, and we measure how much of a cytokine called interferon gamma they secreted. And what you see is that naive mice secrete a lot of interferon gamma, whereas lymph nodes from the tumor-bearing animal don't secrete much interferon gamma at all. And so we asked, could we replicate that on the chip? So we took lymph nodes that were from non-draining nodes from a tumor-bearing mouse, and then coupled them with a tumor and cultured overnight. And for comparison, we took those same lymph nodes, other slices from those nodes, and cultured them with healthy tissue, a fat pad. And we found that here, culture with the tumor, again, suppressed the response of the T cells compared to the healthy fat. So that was really exciting because this is the first indication of immunosuppression on a chip with intact tissue. And moving forward, we are hoping to further test other features of immunosuppression and the interaction between lymph nodes and tissues from tumors in order to see if we have a model here of tumor immunity that could, for example, be used to test new therapies. But this type of model that holds two tissue slices, I would argue, is applicable to other things besides tumor immunity. And so maybe you're having some ideas for your own work. This could be applied to brain immunity or even other non-immune systems, such as the gut-brain axis and other inter-organ interactions that people are interested in. Okay, so just to wrap up, I've talked to you about the combination of ex vivo samples of tissue with microfluidics and how this provides the experimental control that you would get with a cell culture dish, but preserves the spatial structure. The tissues are isolated from other organs in the body, which may be good or bad, depending on what you're trying to ask about. And I've shown you two applications, one in local drug delivery and one in multi-tissue communication. Others have used this type of system for personalized drug screening and for long-term tissue culture and differentiation. And there's lots still to do here. So there's really only a handful of labs in the world that combine intact tissues with microfluidics. And so more work is needed in developing various methods for on-chip analysis of these tissues, particularly with spatial resolution, but also with high content omics type technologies. On-chip long-term culture of tissues is still a major challenge for many tissue types. And then using these technologies by collaborating with the biologists who care about these tissues in order to answer biological questions and develop new therapies for disease. That's a huge outstanding need. And we don't want to just make technologies, right? We want to use them to better human health in this case. Okay, so I'd like to thank my lab. I showed you the people who were doing the work along the way. And I'd also definitely like to thank our collaborator, Professor Jennifer Munson at Virginia Tech, who was instrumental in the tumor immune study that I showed you. We also work with John Lukens and Melanie Ruskowski at the University of Virginia. And this work was generously funded by the NIH and the Hartwell Foundation and a number of excellent foundations. So thank you to them. And I'd like to remind you to just ask questions if you have them, and I'll respond to them by email.